Okay, so this is why we're talking about excitation contraction coupling, which we might not be able to cover contraction until next lecture. But again, it's due to these ion channels and ion movement. This causes disturbances and changes or in the cell's overall electrical charges and balances between the inside of a cell and the outside of a cell. This is something we call membrane potential. For exam 2, don't worry too much about it at this point. Again, exam 3 is where we get a little more into this. But basically what happens, you have these changes in ion concentrations and movement, and this travels all the way to the end of a neuron called the axon terminal. This causes a neuron to release something called acetylcholine, ACH for short. So thing is that acetylcholine triggers electrochemical activity in the muscle cells that are targets of these motor neurons. Remember, a motor unit is a motor neuron connected to a muscle fiber or a group of muscle fibers. So acetylcholine relays the signal from a neuron to a muscle cell. And then this causes a release of calcium into a muscle cell's cytoplasm, also known as the sarcoplasm. And then what's next? So this whole thing is what you talk about, at least in terms of excitation contraction coupling. So all of that chemical movement, remember, all these ions have electrical charges. So this is the excitation part of that when we refer to this term. So the ion movement, all those action potentials, all the movements of changes in positive and negative charges, that's the excitation part. Now we have something called thick and thin filaments. So here's a thin filament, and thin filaments, they're mostly composed of a protein called actin. And in this picture, this kind of like red, yellowish, peachish color, this is actin. These are actin filaments. There are other parts of it, but we'll get to that real soon. Then you have thick filaments, and thick filaments, yeah, they're really thick, and they mostly contain a protein called myosin. So this is my mnemonic. So how do I remember that actin filaments are thin, or thin filaments are mostly actin, thick filaments are mostly myosin? So, or like, I think there's two mnemonics that kind of go hand in hand. I think like, if you've seen like an actor in real life, like I was always really surprised to see like the people who acted on Hawaii Five-0. They're a lot thinner and leaner in real life, but like I guess because like they say the camera adds 10 pounds. So I think if people are actin, they're typically thin. But another mnemonic that goes with that, so actin is thin, and then myosin. So whenever you see it, so this is another thing. You think, if say your friend's been working out and they got really thick, right? So you think, oh my, they are thick. So this is the way you think. Myosin is thick. Oh my, they are thick, or tick if you're local. So yeah, I love this, or I love the neo drop, but yeah. So oh my, they are thick. That's how I remember myosin is a thick filament. Okay, so why do I know about myosin and actin? Well, the thing is that in these, what we have here, thick filaments and thin filaments. And things that these, remember that oh my, they're thick, and not only thick, very muscular as well. So things that myosin is kind of like the muscle part of this contraction. So they're doing the pulling. The actin's kind of along for the ride. So who's going to win? Like a tug of war between very like thin models, or this rugby team that's oh my, they're thick right here. So this, again, the thicker parts are going to win that tug of war. So myosin has something called Myosin Heads that help with thickness and also help to pull these actin filaments. And if you need to go somewhere, I am going to run a little over time. I'm going to try to cover contraction at the very basics with this. So myosin, and remember, myosin, my, they're thick, and then actin, they're actin and they're thin. So things that myosin have these little parts of the myosin, these thick filaments called, or actually myosin has something called these heads, and these are what actually pull actin filaments. And what happens when you pull actin? Well, notice that this muscle is this complex of proteins. Notice that these jagged lines I'm showing right here, and we'll get to that later on in the next lecture. Don't worry about for now. This is what you call Z lines. They denote the borders of something we call sarcomere. But what do you notice between these zigzag lines? They get shorter and shorter and shorter. So myosin is why muscles contracting, they get shorter, because they're pulling on the actin and making it shorten in length. So then what happens here is that, again, ion movement, action potentials. Now we're finally getting to contraction. So what's the purpose of calcium? Did I just want to tell you about chemicals? Well, calcium serves a very important purpose that drives this actual contraction and shortening of these myofilaments. So there's something called troponin and tropomyosin. So here's a thin filament, but there are other parts. You have nebulin in the central core. I'm not going to focus too much on that. But again, here's myosin. But myosin wants to get with actin. Thing is that when a muscle is not contracting, there's something in the way. So what we have is something called tropomyosin. Tropomyosin is actually blocking myosin in this case. So now we have something called active site. If you're taking biochemistry or going to biology, you definitely got to know what active site is. But for now, just to know that active sites are where myosin bind to actin. But tropomyosin is kind of blocking it. So myosin really wants to get with actin, but it's got tropomyosin in the way. And troponin is also helping. So this thing about troponin, I'd like to think of it as like tropomyosin as like a barrier or a velvet rope that prevents actin and myosin from getting together. So you have tropomyosin kind of getting in the way. So how do you get troponin to move tropomyosin out of the way? Well, again, troponin, I like to say, is like the bouncer. Tropomyosin is like the velvet rope. So how do you get the bouncer to let you in from one place to another? Well, here's troponin, here's tropomyosin. Typically, you have to wait in line. But say you have money to spare and the bouncer is easily persuaded, you typically give them something, right? So what you're giving for tropomyosin and troponin is that you're giving calcium. Calcium, when troponin has calcium in hand, what it does is move tropomyosin out of the way. So calcium is kind of like what helps. So what happens? Again, when the muscle is at rest and there's no contraction, troponin and tropomyosin is blocking myosin from binding to actin. But now we have calcium in the equation. Now troponin is happy, and what it does with calcium in hand, it moves tropomyosin out of the way, and now myosin can bind to actin. So troponin isn't doing the blocking itself, but it's tropomyosin that's doing the blocking. But once troponin has calcium, now it can move tropomyosin out of the way. So again, if you have tropomyosin, it's kind of like blocking myosin from binding to actin. So if tropomyosin is blocking myosin and actin from interacting, there's no pulling, there's no contraction. So whenever a muscle is relaxed, you have tropomyosin blocking myosin and actin, or actually wrapping around actin to block myosin from binding to actin. But once you add calcium, remember troponin is the one that kind of moves away myosin from so that's what's happening here. You have all this troponin, and now it's pulling tropomyosin away from the active site where myosin wants to bind to. So now you can actually have myosin pulling actin. So that's why I like to say, like, troponin is the bouncer, tropomyosin is the rope. And I think that's it. So if you want to do some top hat questions for review, let's do that. But again, we're over time, so you don't have to stay for this. All right. So let's see. Hopefully this sunk in. Let's go to top hat. So which filament wraps around actin and blocks myosin from binding? Okay, let's see how everyone did. And then, so it seems like most of you said tropomyosin, some of you said troponin, and the people who said tropomyosin are correct. So tropomyosin is actually the physical barrier that blocks myosin from binding to actin. Troponin is the one that moves it away. So which of these filaments binds to calcium? Actually, I should say proteins. Which of these proteins bind to calcium? I'm kind of like giving you a hint there. Yeah, so which protein binds to calcium? Yeah, which of these proteins, it should say proteins, binds to calcium? Okay, so then which of these proteins? Most of you said troponin, and most of you are correct. Yeah, I should correct that question. Troponin is not a filament, but remember how this is the one that once it has calcium in hand, it pulls back on tropomyosin to reveal the site where myosin will bind to actin. So that's why I say like troponin is like the bouncer. And then thick filaments are mostly made up of, okay, let's see how everyone did. So most of you said myosin and most of you are correct. Let's see. Yeah, so most of you are correct. Let's see. It's not showing how to. So okay, but yeah, most of you are correct. It is it is myosin. So thick filaments are mostly made out of myosin. So remember that mnemonic, my, they are thick. So again, myosin, the primary component of thick filaments, actin, the primary component of thin filaments. You also have troponin and tropomyosin. But again, the myosin and actin is what actually causes the contraction of actual myofibril. Okay, so enjoy your weekend, everyone. Sorry for the technical difficulties earlier, and see you on Monday. Wednesday will be pre-recorded because, again, I have to help out at the med school for their sim lab. Actually, I think it starts at 12, so yeah. It will be up on, so Monday is, yeah, full lecture, but Wednesday we're going to have pre-recorded, all right? Okay, thanks for tuning in, and again, as always, take care and stay healthy.